Hello students, today we are going to start the next polymer of amino acid. So the next polymer of amino acid is protein. So already I have told you that how proteins are formed, right? So the proteins are formed actually by the polymerization of amino acids. So one amino acid attached to the next amino acid by a bond that is called as peptide bond, right? So by this peptide bond, the amino acids are attached to each other. Okay, so this is suppose a long chain of amino acids. So in this long chain of amino acid, we will get amino group on the left hand side, and on the right hand side we are going to have a carboxylic acid group. So in case of this protein, what we can get that the protein is going to have a N terminal and a C terminal. Remember the C terminal, that C indicates the carboxylic acid, and this N terminal, that N indicates amino group. Now we have to understand what is actually a peptide, a polypeptide and a protein. So what is the difference between polypeptide and a protein? Okay, so what is it? If it comes to the polypeptide, it is not functional. Okay, it is a non-functional chain. Okay, but whenever it comes to the protein, it is the functional one. Okay, second thing, in case of this polypeptide, they will have just the linear structure like this. Okay, but in case of the proteins, it can be folded. Okay, like this. So this will be linear, it will be folded. Okay, so to be called as a protein, this polypeptide chain, suppose this is a polypeptide chain, it is not necessary that it can be a protein. So folding have to be there, and not only folding, it can be unfolded, but it has to be functional. It starts to work something. So yes, that is the difference between the polypeptide and a protein. So after that, let us see the characteristics of proteins. What are the proteins actually? Okay, so proteins are the polymers of amino acid. Okay, it is the polymer of amino acid. Second thing, what we have got in the polymer thing, we have already studied in the acid soluble or acid insoluble part, that in this acid insoluble part, we have seen only the three types of polymers. What are the polymers? One is the polysaccharide, then we have seen the nucleic acid, and third one is the protein. So here this protein are found in acid soluble or insoluble pool. It will be always acid insoluble pool because it is going to have molecular weight more than 800 Dalton. Next one, this protein, they are functional and they are performing different function in our body. Okay, like different type of enzymes, okay, different type of carriers, okay, like hemoglobin, they are all protein. So these proteins, they are the one more thing we need to know whenever it comes to the polymer can be of two types. One is homopolymer and another is the heteropolymer. So in case of proteins are always heteropolymer. Remember, heteropolymer means among the 20 amino acids. Okay, so we have the 20 amino acids, essential and non-essential together. So in case of this 20 amino acids can come in number of times. Okay, so that will be a heteropolymer. Okay, so this is a protein. Now the next thing, that in case of these proteins, they have another very important character, that it is a heteropolymer. Already told these are sometimes it is insoluble. Okay, normally it is insoluble in water because it is a acid insoluble structure. Okay, it is found in the acid insoluble pool. Now the type wise, okay, so we don't have to know many about the proteins. Very few things we have, depending upon the coiling or depending upon the complex structures. The protein either can form primary structure, secondary structure, tertiary structure and quaternary structure. Okay, so we have depending upon its arrangement, how it forms, they can form primary structure, secondary structure, tertiary structure and quaternary structure. Yes, we also have another type of protein. It can be fibrous, okay, or it can be globular protein. Okay, so we are going to have different classifications of protein depending upon this. Depending upon the folding, we have primary, secondary, tertiary and quaternary structure, and depending upon solubility. If it is not soluble, it will be fibrous. If it is insoluble, it will be fibrous. If it is soluble, it will be globular. So we have this different types of protein. Now we are going to see the types, first primary, then secondary, then tertiary and then quaternary structure. So let's just see what are the primary, secondary, tertiary and quaternary structure. Okay, so if it is the primary structure, remember what happens. Primary structures are generally is a linear chain. One side it is going to have N terminal and other side it is going to have C terminal. So this will be a linear structure, and here we will get only the peptide chain, not more than that. So this will be called as the primary structure. So these are generally non-functional. Okay, they're generally not functional whenever it is just synthesized in our body. First of all, any kind of protein will attain the primary structure, but later on the primary structure will go on dividing, and after that, not dividing, go on become more condensed to form secondary, and then it will convert into tertiary. This way it will go on changing its structure. So normally they are not functional, but yes, there is one exception also. What is that exception? We have one functional linear primary structured protein, that is actually one hormone, that is growth hormone, right? So growth hormone is found in the primary structure. It is not non-functional, it is functional, because growth hormone perform different functions in our body. It helps us to grow, isn't that? So this will be the primary structure. Normally they are non-functional. Second one, we will see secondary structure. So what happened in secondary structure? Yes, we have two types of secondary structure. One is called as the alpha helix and second one is called as a beta pleated sheet. Okay, so we can have alpha helix and beta pleated sheet. So what is alpha helix now? It is going to have the coiling. So this way there will be coiling. We are going to have always right-handed coil. Okay, it is going to have right-handed coil. Second thing, in this we can have the peptide as well as hydrogen bond. And remember, this is the perpendicular structure, and hydrogen bond will be formed in that same chain. Okay, so this is the same chain in that we can have hydrogen bond. Suppose this way hydrogen bond is formed. Okay, so hydrogen bond will be always always perpendicular to axis. So this is the axis. On the axis, the hydrogen bond will be always perpendicular. Yeah, it is relatively more stable as compared to the primary structure, but it is not even the complex, or it is not completely the stable structure. It is also not that stable. Yes, it is as compared to the primary structure, it is stable. We can have one example. What is that example? Example is alpha keratin. So alpha keratin is found in our hair, in our nail. So this will be called as the alpha keratin. The second one is beta pleated sheet. Earlier in the alpha helix, what we have seen, there is only one single polypeptide. Now in beta pleated sheet, what happens? We have more than one polypeptide, and hydrogen bond is formed in between the different polypeptide chain. So earlier in alpha helix, we have seen the hydrogen bond in the same polypeptide chain. This is the same polypeptide chain. In the beta pleated sheet, what happens? We can see hydrogen bond, but here hydrogen bond is formed in the different polypeptide chains. So this is the basic difference. In alpha helix, hydrogen bond forms in the same polypeptide chain. Okay, so in the same polypeptide chain we can get hydrogen bond. But if it is beta pleated sheet, then hydrogen bond will be there, but it will be in the different polypeptide chain. Okay, so remember this. Yes, it is also the same, that it is going to have the peptide. Obviously in this amino acid chain, in this polypeptide we have the peptide bond, and in between the different peptides we have hydrogen bonds. So peptide and hydrogen bonds are found also in the beta pleated sheet. So yes, we have one example in the silkworm. They have one type of protein that is called as fibrin protein, which is found in the any silk, that silk cloth we get. In the silk cloth we are going to have beta pleated sheet, that is the fibrin protein. So this is the secondary structure. Examples just remember. Then after that, let's just move to the next structure, that is tertiary structure. So what is the tertiary structure now? So what is the tertiary structure here? What we can see, if further folding occurs. Okay, so if further folding occurs, then it will be called as a tertiary structure. So in this tertiary structure, what type of bonds we can see? We can see hydrogen bond. Yes, first of all, the base will be, the skeleton will be formed by the peptide, then hydrogen bond. We can have the ionic bond, we can have the hydrophobic interaction. Okay, then we can have van der Waals interaction, then we can have the covalent bond. Okay, so any type of bonds can be possible in tertiary structure, and most important, yes, disulfide bond. This type of bonds can be seen in the tertiary structure. As compared to the primary and secondary structure, tertiary structure is more stable. Okay, in our body, different enzymes we have. All the enzymes basically have the tertiary structure. So this is suppose a tertiary structure. Okay, in that we can have hydrogen bond, we can have disulfide bond, and here suppose a water molecule is here. With this water molecule, the hydrophobic end will start to react, isn't that? So that will be that will be ionic bond. Then here we have hydrophobic part, here there will be hydrophobic part. In between these two we have hydrophobic interaction, then van der Waals interaction. This type of interactions can be found in the tertiary structure. So whenever we see a tertiary structure, it contains so much that it develops a tertiary, this type of structure. Suppose here this structure will be a wool-like structure. Okay, so this will be tertiary structure. Yes, different type of functional proteins in our body are the enzymes that will attain the tertiary structure. So after that, next structure we will see the last structure. What is this? This will be the quaternary structure. So what is the quaternary structure? If different type of structures are present together, okay, suppose we have primary, secondary, tertiary, quaternary, tertiary structure are found, and they're forming more, or they're having the bonds in between, then it will be called as quaternary structure. Okay, so here one example I can give, that is hemoglobin molecule. So in this hemoglobin molecule, we have the interactions between two alpha subunit and two beta subunits. So in this subunits, they are going to have alpha helix, they have the beta pleated sheet, they have the tertiary structure, and all of them they have the interaction, and that's how the quaternary structure is obtained. One of example is the hemoglobin. So one of example is hemoglobin. Let's just move to the next part. So we have seen the four types of structures: primary, secondary, tertiary and quaternary. So these are the examples. First, primary structure, it is non-functional. One example is the growth hormone, which is only the exception, that is the functional one. Secondary structure, we have seen two types. One is alpha helix, second one is beta pleated sheet. So in this first alpha helix, we have one example, that is the alpha keratin. Second, beta pleated sheet, where we have one example, that is fibroin. Tertiary structure, maximum enzymes are attaining tertiary structure. Then quaternary structure, one example is hemoglobin. Then after that, let's just move to the last part of this whole protein part, that is the different functions. Yes, in the NCERT we have, okay, remember in the NCERT we have one chart, and from this chart question comes, okay, a number of times. Like this question came a number of times, 2019 this function of GLUT4, that question came. So remember, a chart is here. Whatever the chart will be given in the NCERT, I have already told you from first to last that the charts are very important, and what part you have to give more concentration already I have told you. So for this protein from the NCERT, you just see that chart. That is going to be important. So after that, see this function from this chart itself I have told. First one, enzymes. I already have told you that the tertiary structures generally have the uh, enzymes, they are attaining tertiary structure. So first one, trypsin is an example of enzyme. Next one, different defense proteins, the different antibodies. I told you about the antibodies. Antibodies are the amino acid chain attached to the another chain by disulfide interaction, disulfide bond. So different defense protein which fight against the foreign pathogen, that will be the immunoglobulin, which is also a protein. Okay, next one is receptor. Okay, we have different receptors, sensory receptors for taste, smell, for vision itself. We have two types of vision sensory cells. They are iodopsin and rhodopsin. Okay, so not cell, rhodopsin is a protein, iodopsin. Okay, these are the proteins which are available in our eye so that we can able to see the different colors. Okay, sensory proteins that are rhodopsin for the light, dark and white vision. Okay, black and white vision, and for iodopsin we can see a purple pigment. Okay, so these are the receptor proteins. Next on the transport protein, this question came, GLUT4, which is present on the surface, okay, surface of cell, which is helping in transport of glucose inside it. Okay, by the symport, actually glucose and sodium will be transporting from outside to inside by this GLUT4. This is one transport protein found in the cell membrane which helps in transporting the glucose from outside to inside a cell. Next on collagen, which is very much important. Collagen is the most abundant protein in the animal kingdom. Remember that. So collagen is very much important. Next one is conjugated protein. So different conjugated protein like albumin. Okay, albumin is also very much important. Albumin is an example of a conjugated protein. So different type of proteins and their functions we have to see. Remember this thing. And one example we can give, that is Rubisco. Ribulose by bisphosphate carboxylase oxygenase. This is abundant. This is the abundant protein found in biosphere. Okay, so this is called as the Rubisco, which is found in the plant actually, which is helping in photosynthesis process. So it is the most abundant protein in the whole biosphere. And what is the most abundant protein in the animal kingdom? That is collagen, right? So collagen is actually the fiber, white fiber, which is giving us the strength. Okay, so these are the different functions of protein. I hope you understand about the amino acids and proteins. That are all the things I have already covered. So you have to see whatever I have told you in this video. The primary, secondary, tertiary and quaternary structures, they are very important. So that's the NCERT chart from where you have to see what are the different functions of protein. So that's all about the video. I hope you understand. Thank you.